Hi, everybody. So 3 years ago, I was walking on a river in the Northwest Highlands of Scotland. This river, known as the River Carron, had a problem. That problem was that the native sea trout populations were rapidly declining. There had been talk about restoration projects for years, but one man had independently begun restoring the populations by using electrofishing to capture adult sea trout and raise their offspring. He was successful. So I tell this story because I think it demonstrates what's happening internationally to manage salmonid populations. Here in Canada, we manage our fisheries differently, with electrofishing being discouraged in some jurisdictions that operate under the precautionary principle. And these are used to protect spawning. So hello, everyone. My name is Bradley Howell. Today, I'm going to be talking about a project that I started in September of 2020 at Trent University in Peterborough, Ontario, which addresses electrofishing to collect spawning salmonids. I just want to include a quick thank you to those that helped me along the way and inspired me throughout the project. Those people are listed here. And I also want to thank the Peterborough field naturalists who provided some funding for the project. So I'm very excited to share that the work that I'm going to talk about today has just been published in the North American Journal of Fisheries Management. It's titled "Capture of Spawning Brook Trout by Electrofishing Does Not Impair Embryo Survival." So electrofishing is widely used as a tool to complete population and community assessment in freshwater systems. However, using electrofishing as a technique to collect wild fish gametes is much less common. And in Ontario, hatcheries provide support to restoration and reintroduction projects, helping to maintain and restore healthy fish populations. Brook trout are one of the species commonly grown in hatcheries in Ontario from gametes collected from wild fish. Inconsistency in the type of electrofishing equipment used and the level of fish exposure observed in other studies has led to difficulties interpreting results, assessing the effect of electrofishing on gamete quality and viability. While it is currently considered to be a relatively safe method of collection, there are potential risks with its use arising from improper voltage and user inexperience. Negative physical impacts of electrofishing have already been documented in adult fish, including spinal injury, hemorrhaging, and mortality. However, studies examining salmonids and the physical impact of electrofishing, along with its effect on gamete viability, have had conflicting conclusions. So examining brook trout is valuable because size and immobilization threshold may change with species, and brook trout really haven't ever been explored in relation to electrofishing before. So for my project, the underlying question was, does electrofishing spawning brook trout affect embryo survival? I wanted to determine whether electrofishing affects gametes from male and female fish differently, and whether sex-based gamete viability dictated post-shock development of combined gamete pairs. I predicted that electrofishing would negatively affect brook trout larvae, evident by a lower rate of survival through early life developmental stages compared to offspring from non-shocked adults. To better test this question, I examined 2 strains of Ontario brook trout to determine whether domestication had any effect on gamete survival. I accomplished this by exposing both strains of adult brook trout to pulsed DC electrofishing and monitor the development of different embryo treatment families through fertilization, eyed egg, alevin, and fry stages. So my research took place at the Ontario Ministry of Northern Development, Mines, Natural Resources, and Forestry, Codrington Fisheries Research Hatchery. That's a large name. The research hatchery is in Codrington, Ontario, which is about an hour from Trent University, which is located in Peterborough. We also respectfully acknowledge that the work and learning were completed on the treaty and traditional territory of the Mississauga Anishinaabe, and we offer gratitude to the First Nations for their ongoing stewardship of our ecosystems. One parent population was derived from Scott Lake in Algonquin Park, where adults at the Codrington facility were brought in as gametes from a wild spawn collection. The other parent population were from the provincial Hills Lake Hatchery broodstock, which has been in the provincial hatchery system for multiple generations and is considered to be heavily domesticated. Adults from both strains used in this study had been raised at the Codrington facility for 6 years prior to their use in this study. So 60 individual fish were randomly assigned to one of 4 treatments. To determine whether there was any sex-based gamete survival bias post-electroshocking, both males and females were shocked in a fully factorial design, which involved a non-shocked female and a non-shocked male, a shocked female and a non-shocked male, a non-shocked female and a shocked male, and then a shocked female and shocked male. These 4 groups were used to create 58 distinct families, which were composed of 1 female per family and 2 pooled males. And that's what you see with the bracketing here on this slide. So shocked treatment groups were moved to a raceway 2 days prior to treatment and shocked using pulse DC electrofishing for 10 seconds at 102 watts. While control groups remained in separate fiberglass tanks. And so all the fish were held in water, continuously supplied by a flow-through gravity system throughout the experiments, and that drew from the on-site creek. Water temperature remained fairly stable throughout all of the tests, so that was around 9.1 degrees Celsius, and conductivity also didn't fluctuate much with the conductivity of 376.33. Fish were collected with a net after having been shocked or not, and then transferred into an anesthetic bath, and in this case, we used MS-222. So their eggs and milt were then stripped by hand. Once fertilized in containers, the eggs were washed with water to activate them before being placed randomly but evenly into 2 vertical incubation stacks, which are also known as Heath stacks, and then they were separated by family. It's important to note that each family was split between 2 separate incubation stacks to avoid instrumental bias on egg viability, and water was again sourced from the flow-through water system from the on-site creek. Eggs were then examined thrice a week to remove and record dead eggs. This allowed us to look at survival rate over time, and I stopped monitoring development at 3 weeks post-hatch for all of the treatments. So this all happened between October 2020 and March 2021, and I do have to tell you that I saw some really cool mutations over those 3 weeks, including 1 3-headed fry, and that got a lot of Twitter traction, which I was happy about. And so the strain of the fish, i.e. the wild versus domestic, was not a predictor of embryo survivorship, suggesting that wild origin fish did not differ from the domestic strain of brook trout in terms of baseline incubation success or potential reproductive impacts of electrofishing. Kaplan-Meier survival curves comparing the 2 strains have some time-specific differences, such as lower proportion of individuals remaining at the hatch date for the domestic lake strain. That's the second vertical gray line in this figure. But at the end of the experiment, the 2 survival rates were comparable, with the domestic strain having a survival rate of 0.76 and the wild origin having a survival rate of 0.78. So what we're seeing with these survival curves are the proportional survival of all the offspring on the Y over time on the. So the previous slide showed survival between strains, and so now we're looking at survival between the treatments. So the survival curves are a little misleading because when you take a look at the embryo proportional survival between the 4 treatments, it looks as though there were some noticeable differences. The most important thing to notice is that in the treatment where we shocked both male and female fish, the survival rate was actually the highest with a survival rate of 0.87. And this seemed really exciting, but later on, we found that our statistical analysis debunked this. And so in our beta regression models, we did see some statistical differences, but again, they were only between the male shocked and female shocked groups in one of the Heath stacks. So here, the significant proportional survival differences are circled in red. Again, we see proportional survival on the Y and treatment on the X. And so the shocked male and shocked female treatment had 15.5% higher survival in the control treatment. However, these differences were diminished to the point of statistical insignificance when survival data from both Heath stacks were combined. And so we use beta regression model fit with AIC, and pairwise treatments were conducted with Kruskal, Wallace, and Duns. And there was not a sex-based impairment of offspring survival following electrofishing. Because we didn't test for family effects, in other words, the genetic combination of the parental fish, and because we exposed parental fish rather than the embryos itself, it is likely that the pseudo-significance that we observed is likely from a few lower performing families being assigned to those treatments simply by random chance. So just some quick things to note. We did use pulse DC electrofishing, which has been shown to be less harmful than other forms of electrofishing, such as AC. We used low electrofishing settings, but those may have increased fish exposure because we shocked them for 10 seconds, regardless of whether or not they had lost equilibrium. So they might have been exposed to electrofishing more than what they would typically be exposed to in an in-stream electrofishing capture. So just some management implications. Apparently, we can shock fish and it won't impact the survival of those offsprings. So because of that, gamete collection could be an exception to the current protection that we have over fall spawning. Other possible negative environmental effects like red disturbance could still occur during that spawning period. So for reasons other than gamete collection, we probably would still continue to discourage electrofishing. Thanks.